Okay, let's unpack this. The idea of actually uh, engineering the extinction of a species. It sounds like something straight out of a sci-fi movie, doesn't it? But that's precisely the territory we're exploring today.
It is. It's interesting because, so much of conservation is focused on saving species, right? But now we're looking at these really complex questions around, well, intentionally ending a species using cutting-edge tools,
Yeah.
genome modification specifically.
Exactly. And for this deep dive, you shared a really compelling paper, Deliberate Extinction by Genome Modification, an ethical challenge.
Mm.
So our goal today is sort of navigate through this complex area, look at some concrete examples, and really try to grasp the different perspectives in this debate. Think of it as your shortcut to understanding this whole discussion.
And it immediately brings up a core tension, does it? I mean, how do we square our strong commitment to biodiversity with the possibility that maybe in certain very specific situations, deliberately causing extinction might be justified? It's a real paradox.
Yeah, a paradox right at the heart of it. And the paper really highlights this by looking at three specific scenarios where this idea, using genome modification for eradication, is actually being considered. The first one that really jumped out at me was the New World screwworm.
Ah, the screwworm, yes. It's a particularly unpleasant parasitic fly. Its larvae, they infest warm-blooded animals, livestock, wildlife, and yeah, sometimes even humans. That's where the name hominivorax comes from.
Oof, yeah.
And this infestation, it causes severe suffering. It can lead to death, often from secondary infections. The fact that it's parasitic, relying directly on a host, that often factors into how people think about these cases compared to, say, free-living species.
Gruesome indeed. But what's interesting is that we've actually had some success in controlling it already, haven't we?
Yeah.
Using the sterile insect technique, SIT.
That's right.
So even though SIT is a kind of genetic control we've sort of accepted, does the intentional design part of these newer methods,
Hmm.
does that raise different concerns for people? Is there like a psychological thing about engineering extinction that maybe wasn't there with just sterilizing insects?
That's a really key question. SIT has been, well, a valuable tool used in North and Central America for decades now. It involves mass rearing screwworm pupa, sterilizing the adult flies with radiation, and then releasing them. They mate with wild flies, but no viable offspring, so the wild population drops.
Right. So it is a form of genetic population control, just maybe not as targeted as what's being discussed now.
Hmm.
The paper talks about how genome modification could actually make SIT much better.
Yeah, enhance it. And one really interesting aspect is the potential for these much more targeted approaches. So one method, it involves genetically modifying the screwworms so only the female larvae need tetracycline, an antibiotic, to mature, which means researchers can just eliminate the females during rearing. And that has several advantages. It lowers the costs of rearing them, for one. Plus, you can use less radiation on the males, which might make them healthier, more effective at mating out there. And it ensures only sterile males, the ones that actually cause the population decline, are released.
That sounds really efficient, streamlining the whole thing and making it work better. And the paper also mentioned more advanced techniques like FSRDL and gene drives.
Correct. FSRDL, that's female-specific release of insect with a dominant lethal. That would involve releasing modified males that pass on a gene that specifically kills off female offspring.
Wow.
And gene drive technology, well, that goes even further. The aim there is to ensure a specific trait, like female sterility, gets inherited by almost all offspring, generation after generation. That could potentially lead to really rapid eradication.
So the case for getting rid of the screwworm, it seems pretty strong. You've got the immense suffering it causes, the impact on livestock, food supply, how hard infections are to treat. And now with these tools, maybe a permanent solution is actually possible, especially since it's apparently been showing up again recently.
Right, the argument is heavily weighted towards alleviating that suffering and protecting food resources.
But even for something like the screwworm, which causes so much harm, are there any voices raising concerns, maybe about the principle of it, unforeseen ecological effects or the whole slippery slope idea?
Well, if you connect it back to the motivation, it's mostly about the suffering and food security. And while some might argue for the screwworm's intrinsic value, its inherent right to exist, it's a difficult argument to make stick given it's a parasite. Plus, current research suggests its ecological role is, well, pretty limited. It doesn't seem to be a vital part of the ecosystem.
Right. Okay. So it sounds like there's pretty broad support for tackling the screwworm, at least locally. The next case, though, the Anopheles gambiae mosquito, the one that carries malaria. That feels like a much heavier, more ethically complex discussion.
Absolutely. The human toll of malaria is just staggering. Hundreds of thousands of deaths every year, mostly young children in sub-Saharan Africa,
Hmm.
and the economic burden on affected regions is huge. So, yeah, the idea of using genome editing, maybe gene drives to crash mosquito populations by messing with the sex ratio, it's being actively explored.
The paper does mention the risk, though, even if it's considered slight, of potentially wiping out Anopheles gambia
Mhm.
completely, and maybe even other related mosquito species. That's a big potential
It
consequence.
is.
With the screwworm, the argument felt very focused on minimizing suffering, mostly animal suffering. Now, with the Anopheles mosquito and malaria, we're talking immense human suffering, but also a species that's, part of a bigger, maybe more complex ecosystem. How does that change the calculation?
That really brings up a critical question, doesn't it? Does the profound human suffering caused by malaria outweigh the potential loss of these mosquito species? From a purely human-centered perspective, the argument for getting rid of the vector is undeniably strong.
But, as the paper also points out, we don't necessarily have to eliminate the mosquitoes entirely to get rid of malaria, do we? There are other things. Bed nets, better healthcare, vaccines are coming along, and even gene drives that just stop the mosquito from transmitting the malaria parasite itself.
Precisely. And we have had successes in the past controlling malaria, insecticides, changing mosquito breeding habitats. So while maybe temporary local suppression of Anopheles gambiae might be needed in some areas, there isn't really widespread support for total global extinction of the mosquito itself.
Mhm.
An interesting alternative, though, is the idea of targeting Plasmodium, the actual malaria parasite, using genome modification. Now, that would also be deliberate extinction, but targeting the disease organism itself, many might find that ethically easier to justify.
That makes sense. Going after the parasite causing the harm versus the entire species that carries it, even if it does carry it. Okay, so the third case study shifts again, this time to invasive species, the house mouse, black rat, Norway rat, specifically on islands in Oceania.
Yes, these small mammals, they seem common, but where they've been introduced on islands, they've had a devastating impact, particularly on native bird populations. They eat eggs, they eat the young. They're driving unique island species towards extinction.
And the idea here is using these sex-biasing gene drives again, but this time as a maybe more humane, more targeted way to control them, compared to the usual methods like traps and poisons, which can affect other wildlife too.
Exactly. The potential for a gene drive to spread through just the invasive rat or mouse population, skew the sex ratio, make the numbers dwindle, it offers a much more species-specific tool. However, and the paper really highlights this, there's that small risk. It's not zero. The risk of global extinction if some of these modified animals somehow got off the island, and the gene drive actually worked across the whole species.
Right. And even though these rodents are seen as pests and they're causing extinctions themselves, the paper says there's general agreement against trying local extinction if there's any real risk of it going global. That feels like a really important line to draw.
It is. It's a significant ethical boundary, and researchers are actively working on ways around it, like developing gene drives that only work for a limited time or only in specific subpopulations to try and reduce that risk.
So, stepping back a bit, looking at the bigger picture for conservation, these examples show how precise these genomic tools can be, right? Compared to older methods that were much broader.
Definitely. And the very idea, the possibility of deliberate extinction, it really forces us to examine how we value species in the first place, both their inherent worth and maybe their practical value or disvalue to us.
You mentioned intrinsic value earlier. For listeners, does that basically mean a species has value just because it exists, regardless of whether it helps or harms us?
That's a good way to think about it, yeah. But while Western ethics often talks about this idea, this equal intrinsic value for all species, our willingness to even consider eradicating pests or disease vectors suggests that in practice, we don't always treat that value as absolute.
Hmm.
It seems other factors might creep in, like whether a species is parasitic or just our general feeling about where it fits in nature, even if we don't consciously admit it.
It's almost like a modern version of that old great chain of being idea, isn't it? Where some species were seen as higher or better than others. The paper mentions that idea, while largely rejected by scholars in the West, might still resonate in some other philosophical
Yeah,
traditions.
precisely. And it makes you wonder, right? Do we really value a Plasmodium parasite the same way we value the mosquito carrying it, or human being? The paper also raises an interesting point about how we perceive the technology itself, genome modification.
Yeah, it notes how the public generally accepted SIT for ages, even though it is fundamentally about using genetic changes to suppress populations.
Right.
But there seems to be more pushback, more apprehension about the idea of more precise genome editing.
Right.
So even with SIT being accepted,
Hmm.
are these newer genetic tools making us rethink our comfort level, our comfort with intentionally manipulating life at such a fundamental level?
Well, if you look at the mechanism underneath, both SIT and genome modification use genetic changes to suppress populations. Maybe the perceived difference is this idea of human design, that the direct control we have with genome editing feels, I don't know, more interventionist than the sort of less direct radiation used in SIT.
But as the paper argues, even SIT irradiation is controlled.
Mm.
It's done to achieve a specific outcome, sterilization. It's not just random zapping.
No, absolutely. It's a controlled process.
So it does raise the question, if we accept SIT, which is a form of genetic modification, why wouldn't we be open to potentially more efficient, more targeted genetic modifications like FSRADL or gene drives if the end goal is the same? Which brings us neatly to the governance side of things. How do we make these decisions?
Exactly. And the paper is very clear. There are no simple, universal rules for deciding when deliberate extinction via genome editing might be okay.
It really boils down to weighing different values, doesn't it? And recognizing that different cultures, different communities are going to have different perspectives, different priorities,
Yeah.
which makes the whole decision-making process incredibly complex.
Absolutely. The paper really emphasizes the need for proper collective decision-making processes. That means getting input from all the relevant stakeholders, upholding democratic legitimacy. And a really crucial piece here is the role of local populations.
Right, the idea that the people who are going to be most directly affected, if a genetically modified organism is released in their backyard, they should have the primary say. That seems fundamental, especially considering their local knowledge and the social and ecological impacts.
And importantly, the paper stresses the distinct rights of indigenous peoples, their right to self-determination regarding their lands and resources, giving them a primary role in these decisions too. Prioritizing local input, it can also be a way to, perhaps, start fixing some historical wrongs in environmental policy, especially in post-colonial situations where local communities were often shut out of decisions affecting them.
That's a really important point. The paper also warns about the sort of dominance of the global north in developing and governing these technologies, making sure the voices from the global south aren't ground out, especially when decisions affect their environments.
And there's a key distinction made between efforts aimed at local extinction versus global extinction. For local efforts, local preferences should probably weigh very heavily, assuming the risks outside that area are low. But the prospect of full global extinction, that might challenge the idea that only local preferences matter.
Because everyone has a stake then.
Well, potentially. Distant populations also have a stake in the planet's shared ecological heritage in international conservation efforts.
But ultimately, the paper makes a really strong case for equal environmental decision-making freedoms. And it concludes that we need a strong collective approach when we're talking about the very existence of a species.
Right. So to kind of wrap up, we've done this deep dive into the really thorny ethical questions around deliberately wiping out a species using genome modification. The case studies, the screwworm, the malaria mosquito, invasive rodents, they show just how nuanced this is, balancing potential benefits against the value we place on species. We've also touched on how our view of a species' value can be shaped by different things, and just how critical, inclusive, fair governance is going to be for making these huge decisions.
Exactly. So if you, listening, were looking for a thorough but hopefully accessible way into this major, fast-moving debate,
Yeah.
well, you've definitely got a handle on it now.
Which leaves us with maybe a final thought for you to mull over. Given how powerful these genomic technologies are and all the different values and viewpoints involved, what fundamental principles do you think should guide us when we make decisions about intentionally changing or even eradicating a species? It's a question that's only going to get more pressing as these technologies keep developing.